Okay, now we have this question and they gave us again two genotypes. Here there are the two genotypes. This is the one, the first one, and this is the second one. And as you can see here, these two genotypes, in some cases, some of the genes, there are four genes, and some of them are homozygous, some of them are heterozygous. And the idea is that they're asking us when you crosses, uh, what is the proportion of them that will be homozygous dominant? In other words, what is the proportion that they have, that they will be AA, BB, CC, all capital, and D. So this look complicated, but it's actually pretty easy. So the first thing that you have to do is to see the proportions that when you have the first gene, here you have this one, this is the gene A, and here you have the other gene. And when you do this, when you make the cross, so here I've made the Punnett square, and here I did the assortment of the alleles. And as you can see here, after I finish the Punnett square, the proportion that all of them are homozygous, here are only half. Yeah, the other ones will be, the other half will not be homozygous dominant. Yeah, now we do the same thing with the other gene. So it's pretty easy. You can start doing it when I delete this. Okay, I'm going to do it this time with green. And here you have, here is this one, and here you have a heterozygous again. So now you make your Punnett square, and different from the previous one, most of them here will be or homozygous recessive, that doesn't work, or heterozygous, that are not the one that we're looking for. So here we only have 1/4 of them. And in the same way that we did with this gene, we can do it with C, and then we can do it with D. And I already did it for you. And as you can see here, here are the answers. So half in this case will be half. In the case of the gene B will be 1/4, and in the case of gene will be also 1/4. Yeah. So in other words, it will be some of them. So it will be 1/4 raised to the power of 2 because you have two of them, and one and 1/2 raised to the power of 2. So as you can see, the answer will be this one. It will be none of these options. And why? Because here, as you can see, as we have some cases, for example, AA, you have a homozygous, and you can have to cross it with a heterozygous. While in the case B, you will have a heterozygous, and you have to cross it with a heterozygous. You will just have different proportions, so you will never get. In this case, you will never get this one, or you will never get this one. Why? Because you're raising to a power of 4, so in other words, you're repeating the same type of procedure for the same one. So, and as you can see, it's different for each one of these ones, as I just showed you in the previous slide. So definitely it will not be C or it will not be D. The answer will be this one. So it's none of these options. I'm not asking you to multiply this. You don't need a calculator. I just want that you show me that you understand how to do this exercise and what is the procedure. If you have any more questions, just let me know. Bye.